Dear students, welcome to the massive open online course on human genetics. Today we are going to discuss the topic DNA repair and recombination. The main objectives of this module are to know about the DNA repair, to know about the mechanisms involved in DNA repair, and to know about the DNA recombination. Dear students, let us start with the introduction. Living organisms have the crucial task to preserve their genome and faithfully transmit it across generations. Transmission of genetic information is constantly in a selective balance between the maintenance of genetic stability versus elimination of mutational change and loss of evolutionary potential. Because DNA is the repository of genetic information in each living cell, its integrity and stability are essential to life. The DNA molecule is continuously under the attack of a multitude of exogenous and endogenous genotoxic insults, and it has been estimated that every cell per day experiences up to 10 raised power 5 spontaneous or induced DNA lesions. Although DNA is a highly stable and versatile molecule, but sometimes the damage is caused to it, but it is still able to maintain the integrity of information contained in it by the DNA repair mechanism. The most frequent sources of damage to DNA are the inaccuracy in DNA replication and chemical change in DNA. Malfunction of the process of replication can lead to incorporation of wrong bases which are mismatched with the complementary strand. The damage causing chemicals break the backbone of the strand and chemically alter the composition of the bases. Alkylation, oxidation, and methylation cause damage to the nitrogenous bases in DNA. X-rays and gamma radiations cause single or double-stranded breaks in DNA. Endogenous damage can result from DNA-based lesions like hydrolysis, deamination, depurination, and depyrimidination, and alkylation or oxidation by intracellular free radical oxygen species or ROS that can occur as byproducts of mitochondrial respiration. Mutations can also arise during normal cellular metabolism, for instance by erroneous incorporation of deoxyribonucleotides or DNTPs during replication itself. Environmental sources of damage can be physical like ultraviolet light, ionizing radiations, and thermal disruption, or chemical like chemotherapeutic drugs, industrial chemicals, and cigarette smoke. Their effects vary from agent to agent. For example, ultraviolet light exposure causes the formation of cyclobutane pyrimidine dimers and pyrimidine 6,4-pyrimidone photoproducts. For example, infrared treatment causes the introduction of single and double DNA strand breaks. Chemotherapeutic agents, on the other hand, or chemotherapeutic drugs cause the inter and intrastrand DNA crosslinks. So DNA lesions can alter the primary structure of the double helix, thereby affecting the transcription and replication processes. Erroneous repair of lesions can lead to mutations in the genome that can be inherited to daughter cells from the parents, with deleterious consequences for individuals' health. As a consequence, eukaryotic cells have evolved a complex signaling network of repair processes known as the DNA damage response, or DDR. The importance of DNA repair mechanisms is highlighted by the existence of many devastating human syndromes that are caused by defects in DDR genes. Notably, many of these mutations generally display increased sensitivity to DNA damaging agents and predispose to the development of specific type of cancer. DNA repair involves any of the several mechanisms by which a cell maintains the integrity of its genetic code. Therefore, DNA repair can be defined as a collection of processes by which a cell identifies and corrects damage to the DNA molecules that encode its genome. So, DNA repair ensures the survival of a species by enabling parental DNA to be inherited as faithfully as possible to offsprings. The perpetuation of genetic material from generation to generation depends upon keeping the rates of mutation at low level. DNA has many elaborate mechanisms to repair any damage or distortion caused during DNA replication. DNA repair can be divided into a set of mechanisms that identify and correct damage in DNA molecules. There are two general classes of DNA repair: the direct reversal of the chemical process generating the damage and the replacement of the damaged nucleotide bases. DNA encodes the cell genome and is therefore a permanent copy of a structure necessary for the correct functioning of the entire cell. Change to the structure of DNA can cause mutations and genomic instability, leading to the cancer. So, damage to DNA is caused by the incorporation of incorrect nucleotide bases during DNA replication and the chemical change caused by spontaneous mutations or exposure to environmental factors such as radiations. Dear students, let's now discuss the major classes of DNA repair processes. These are reversal of damage, nucleotide excision repair, base excision repair, mismatch repair, recombinational repair, and error-prone repair. Many of these processes were first studied in bacteria such as Escherichia coli. However, only a few are limited to this species. For instance, nucleotide excision repair and base excision repair are found in virtually all organisms, and they have been well characterized in bacteria, yeast, and mammals as well. Like DNA replication itself, repair of damage and misincorporation is a very old process. Let's now discuss the reversal of damage. Some kinds of covalent alterations to bases in the DNA can be directly reversed. This occurs by specific enzyme systems recognizing the altered base and breaking bonds to remove the adduct or change the base back to its normal structure. Let's now discuss the photoreactivation. Photoreactivation is a light-dependent process used by bacteria to reverse pyrimidine dimers formed by ultraviolet radiation. The enzyme photolyase binds to a pyrimidine dimer and catalyzes a second photochemical reaction, this time using visible light that breaks the cyclobutane ring and reforms the two adjacent thymidylate in DNA. Note that this is not formally the reverse of the reaction that formed the pyrimidine dimers, since energy from visible light is used to break the bonds between the pyrimidines and no ultraviolet radiation is released. However, the result is that the DNA structure has been returned to its original state prior to damage by the ultraviolet light. The photolase enzyme has two subunits, which are encoded by the PHRA and PHRB genes in Escherichia coli. A second example of the reversal of the damage is the removal of methyl groups. For instance, the enzyme methyltransferase O6 encoded by the ADA gene in Escherichia coli recognizes O6 in duplex DNA. It then removes the methyl group, transferring it to an amino acid of the enzyme. The methylated enzyme is no longer active. Hence, this has been referred to as suicide mechanism for the enzyme. So now let us discuss the accession repair. Accession repair is the general mechanism by which repairs are made when one of the double helix strands is damaged. The non-defective strand is used as a template with the damaged DNA on the other strand removed and replaced by the synthesis of new nucleotides. There are three types of accession repair. First, the base accession repair. Base accession repair system involves an enzyme called N-glycosylase, which recognizes the abnormal base and hydrolyzes glycosidic bond between it and the sugar molecule. Another enzyme, an endonuclease, cleaves the DNA backbone on the 5' side of the abnormal base. Then the DNA polymerase, by its exonuclease activity, removes the abnormal base. DNA polymerase then replaces it with normal base, and DNA ligase seals the region. It involves at least 6 enzymes called DNA glycosylases. Each enzyme recognizes specific bases and removes them from DNA strand. The enzymes remove deaminated cytosine, deaminated adenine, alkylated or oxidized base. Base accession repair pathways start with a DNA glycosylation. For example, the enzyme uracil DNA glycosylase removes the uracil that has wrongly joined with guanine, which is really deaminated cytosine. Then AP endonuclease, that is apurinic or a pyrimidinic site, and phosphodiesterase removes sugar phosphate. AP sites arise as a result of loss of a purine or a pyrimidine nitrogenous base. A gap of single nucleotide develops on the DNA, which acts as a template primer for DNA polymerase to synthesize DNA and fill the gap by DNA ligase enzyme by joining the two. Dear students, let's now discuss the second system, that is nucleotide accession repair. Nucleotide accession repair is a widespread mechanism for repairing damage to DNA and recognizes multiple damaged bases. Nucleotide repair system includes 3 steps: incision, accession, and synthesis. Incision is done by endonuclease enzyme precisely on either side of the damaged patch of the DNA strand. In this way, damaged portion of the strand is cleaved. So, endonuclease enzyme involved are UVRA, UVRB, which recognize the damaged stretch of the strand. UVRC makes 2 cuts, that is incisions, on either side. So, exonuclease removes the damaged strand. Enzyme involved is UVRD. Later on, DNA polymerase synthesizes the new strand by using complementary strand as a template. DNA ligase forms phosphodiester bond, which seals the ends of newly synthesized strand to make it one complete strand. Dear students, let's now discuss the third type, that is mismatch repair. So, DNA mismatch repair, or MMR, is an inner cellular system of recognizing and repairing DNA replication and recombination errors which have occurred in DNA. That is, MMR is the system that is specifically for repairing copying and combination errors. In order to do this, the DNA mismatch repair machinery or mechanisms are able to distinguish the parental strand of DNA which acted as a template for the synthesized copy or daughter strand. This describes MMR, that is mismatch repairing system, as a strand specific, unlike base excision repair system and nucleotide excision repair system, which can occur on either strand of the DNA. The errors that the mismatch repair mechanism targets are called point mutations. These mutations occur on daughter strand of DNA replicated and include errors where a single nucleotide base is either mistakenly added or deleted or changed. So the enzymes that catalyze mismatch repair system are responsible for this final degree of accuracy of DNA replication. They recognize misincorporated nucleotides, excise them and replace them with the correct nucleotides. In contrast to nucleotide excision repair system, mismatch repair does not operate on bulky adducts or major distortions to the DNA helix. Most of the mismatches are substitutes within a chemical class, that is a C or cytosine incorporated instead of a T, that is thymine. This causes only a subtle helical distortion in the DNA, and the misincorporated nucleotide is a normal component of the DNA. The ability of a cell to recognize a mismatch reflects the exquisite specificity of mut-S, which can distinguish normal base pairs from those resulting from misincorporation of the bases. Of course, the repair machinery needs to know which of the nucleotides at a mismatch pair is the correct one and which was misincorporated. It does this by determining which strand was more recently synthesized and repairing the mismatch on the nascent strand, that is the newly synthesized strand. In Escherichia coli, the methylation of adenine or A in a GATC sequence motif provides a covalent marker for the parental strand. Thus, methylation of DNA is used to discriminate parental from progeny strands. Recall that the DAM methylase catalyzes the transfer of a methyl group to adenine of the pseudo palindromic sequence GATC in duplex DNA. Methylation is delayed for several minutes after replication. In this interval before methylation of the new DNA strand, the mismatch repair system can find any mismatch and direct its repair activity to nucleotides on the unmethylated, newly replicated strand. Thus, replication errors are removed preferentially first. The enzyme complex MUTH, MUTL, and MUTS or MUTHLS catalyzes mismatch repair in Escherichia coli. The genes that encode these enzymes are MUTH, MUTL, and MUTS, and were discovered because strains carrying mutations in them have a high frequency of new mutations. This is called a mutator phenotype, and hence the name MUT was given to these genes. Not all mutator genes are involved in mismatch repair. For example, mutations in the gene encoding the proofreading enzyme of DNA polymerase III also have a mutator phenotype. This gene was independently discovered in screens for defects in DNA replication, that is DNA Q, and mutator gene MUTD. Three complementation groups within the set of mutator alleles have been implicated primarily in mismatch repair. These are MUTH, MUTL, and MUTS. The MUTS will recognize 7 of the 8 possible mismatched base pairs except for the cytosine to cytosine and bind at that site in the duplex DNA. MUTH and MUTL with ATP bound then join the complex, which then moves along the DNA in either direction until it finds a hemimethylated GATC motif, which can be as far as few thousand base pairs away. Until this point, the nucleus function of mut H has been dormant, but it is activated in the presence of ATP at a hemimethylated GATC sequence. It cleaves the unmethylated DNA strand, leaving a nick 5' to the guanine on the strand containing the unmethylated GATC. That is the new DNA strand. The same strand is nicked on the other side of the mismatch. Enzymes involved in other processes of repair and replication catalyze the remaining steps. The segment of single-stranded DNA containing the incorrect nucleotides is to be excised by UVRD, also known as helicase second and MUTU. SSB and exonuclease 1 are also involved in the excision. As the excision process forms the gap, it is filled in by the concerted action of DNA polymerase III. Translation DNA synthesis. Translation DNA synthesis, that is TLS, is the process by which cells copy DNA containing unrepaired damage that blocks progression of the replication fork. DNA polymerase can skip past a lesion on the template strand, leaving behind a gap. It has another option when such a lesion is encountered, which is to synthesize DNA in a non-template directed manner. This is called translation synthesis, bypass synthesis, or error-prone repair. This is the last resort for DNA repair, that is, for example, when repair has not occurred prior to the replication. In translation replication, the DNA polymerase shifts from template-directed synthesis to catalyzing the incorporation of random nucleotides. These random nucleotides are usually mutations, that is in 3 out of 4 times. Hence, this process is also designated error-prone repair. Translation synthesis uses the products of the UMUC and UMUD genes. Dear students, this was all about DNA repair mechanism. Now let's discuss the recombination phenomena. Recombination was first recognized as the primary mechanism through which variation is introduced into the population. Recombination takes place during meiosis when maternal and paternal genes are regrouped in the formation of gametes, that is the sex cells. Recombination occurs randomly in nature as a normal event of meiosis and is enhanced by the phenomena of crossing over, in which gene sequence called linkage groups are disrupted, resulting in an exchange of segments between paired chromosomes that are undergoing separation. DNA damage are repaired by a recombination mechanism known as sister strand exchange. Dear students, let's now discuss the mechanism of recombination. Breakage and reunion. Two homologous duplex of chromosome lying in paired form breaks and rejoin crosswise and recombinants. Breakage and copying. One helix of paired homologous chromosome breaks in 2 segments and one of them is replaced by a newly synthesized segment copied from other helix of paired chromosome. The complete copy choice. According to this theory, a portion of one parental strand of homologous chromosome acts as template for synthesis of copy of its DNA molecule. The process of copying shifts to another parental strand. Thus, recombinants contain some genetic information of one parental strand and some of the other strand. Types of recombination. Recombination is divided mainly into 2 categories: homologous recombination and non-homologous recombination. Dear students, let's discuss homologous recombination first. Homologous recombination, also called as generalized recombination, involves exchange of precisely corresponding sequences between homologous DNA duplexes. It's a reaction between two duplexes of DNA, and the enzyme responsible for this can use any pair of homologous sequences as substrates. Homologous recombination occurs naturally in eukaryotic organisms, bacteria, and certain viruses, and is a powerful tool in genetic engineering. In eukaryotes, homologous recombination occurs during meiosis, playing a critical role in the repair of double-stranded nicks in DNA and increasing genetic diversity by enabling the shuffling of genetic material during chromosomal crossover. In bacteria, homologous recombination is a major mechanism of DNA repair and facilitates the incorporation into DNA of genetic material received via horizontal gene transfer and transformation. In viruses, homologous recombination helps shape viral evolution. Homologous recombination plays essential roles in the repair of DNA double-strand breaks and collapsed replication forks. The fundamental reaction in homologous recombination is the exchange of strands between a single-stranded DNA and a homologous double-stranded DNA. This reaction is carried out by the Rec-A or recombination A or Rad51 family of ATPases. In repair, the double-strand break is resected to produce single-stranded DNA, which is then coated by Rec-A or Rad51 to produce a presynaptic nucleoprotein filament, essentially the substrate-bound form of the recombinase enzyme. This filament then binds to double-stranded DNA to form a synaptic filament, a key intermediate that mediates the search for homology, and when homology is detected, exchange the DNA strands to produce a new double-stranded DNA termed heteroduplex. Dear students, now we will discuss the models for homologous recombination. The first one, the Holliday model. In 1964, Robin Holliday proposed a model that accounted for heteroduplex formation and gene conversion during recombination, although it has been replaced by the double-strand break model, at least for recombination in yeast and higher organisms. It's a useful place to start. It illustrates the critical steps of pairing of homologous duplexes, formation of a heteroduplex, formation of a recombination joint, branch, migration, and resolution. The steps in the Holliday model are two homologous chromosomes, each composed of a duplex DNA, are paired with similar sequences adjacent to each other. An endonuclease nick at corresponding regions of homologous strand of the paired duplexes. The nick ends dissociate from their complementary strands, and each single strand invades the other duplex. This occurs in a reciprocal manner to produce a heteroduplex region derived from one strand from each parental duplex. DNA ligase seals the nicks. The result is a stable joint molecule in which one strand of each parental duplex cross over into the other duplex. This X-shaped joint is called a Holliday intermediate or chi structure. Branch migration then expands the region of heteroduplex. The stable joint can move along the paired duplexes, feeding in more of each invading strand and extending the region of heteroduplex. The recombination intermediate is then resolved by nicking a strand in each duplex and ligation. Now, let us discuss the Meselson and Redding model. Meselson and Redding in 1975 proposed a model and is a more satisfactory scheme than the Holliday model. The Michelson-Rading model of genetic recombination was devised to allow the formation of heteroduplex DNA on one chromatid or on two chromatids within the same recombination event. Asymmetrical, that is, one chromatid heteroduplex is formed by a strand displaced by a DNA synthesis from a nick invading a homologous molecule. Here, a single-stranded nick occurs in just one of the double helices. Let's now discuss the double-stranded break model. According to this model, a double-stranded break in one DNA molecule initiates the process of recombination. Double-strand break model of recombination explains production of crossover and non-crossover gametes. So double-strand break recombination results in two Holliday junction. Dear students, let's now discuss the non-homologous recombination. Maintaining genomic integrity is imperative for the survival of an organism. Among different DNA damage, double-strand breaks are considered as most deleterious since they can lead to cell death if left unrepaired or chromosomal rearrangements when misrepaired, leading to cancer. Non-homologous DNA end joining or NHEJ and homologous recombination, that is HR, are the major double-strand break repair pathways in higher eukaryotes, including the humans. HR, being a precise mechanism, uses extensive homology, while NHEJ is error-prone as it utilizes no or limited homology. NHEJ is a quick fix mechanism and operates throughout the cell cycle. During NHEJ, Ku protein heterodimer is recruited to DNA ends followed by DNA PKCs in association with ARTEMIS, which processes double-stranded breaks. Polymerase 1 and or epsilon fills these ends when required, followed by ligation using XLFXR-CC4. DNA ligase 4 complex, also called as specialized recombination, is a pathway that repairs double-stranded breaks in DNA. Non-homologous end joining or NHEJ is a pathway that repairs double-strand breaks in DNA. NHEJ is referred to as non-homologous because the break ends are directly ligated without the need for a homologous template, in contrast to homology-directed repair, which requires a homologous sequence to guide the repair system. NHEJ is typically guided by short homologous DNA sequences called microhomologies. These microhomologies are often present in single-stranded overhangs on the ends of double-strand breaks. When the overhangs are perfectly compatible, NHEJ usually repairs the break accurately. Now let us discuss the types of non-homologous recombination. One is site-specific recombination. Site-specific recombination is a term used to describe a group of processes in which DNA molecules are rearranged by breaking and rejoining the strands at specific points. Site-specific recombination involves two short DNA sequences or sites, which may be within the same molecule or in different molecules. A specialized recombinase enzyme recognizes the sites and then promotes a rearrangement of the DNA such that the left end of one site becomes joined to the right end of the other site and vice versa. This rearrangement requires recombinase catalyzed breaking and rejoining of both DNA strands in each site. A characteristic feature of classical site-specific recombination is that the strand breaking and rejoining reactions are conservative. That is, they do not require any DNA synthesis or degradation, and no enzyme cofactors are required. Transposition. Transposition is a highly specialized form of recombination in which a segment of DNA moves from one location to another location, either on the same chromosome or on a different chromosome. So transposable elements or transposons can be described as discrete DNA segments that are able to move between different non-homologous genomic loci. Transposition of an element is thus a recombination reaction involving three separate sites, the two transposon ends and the new target locus. In autonomous transposable elements, the transposase is encoded by a gene located within the element. Transposon ends often contain inverted repeated sequences on which the transposase binds specifically. Although most transposons can transpose into many different places, insertion is never totally random. While a specific target consensus sequence has been found for some elements, the insertion of many transposons is also influenced by regional features of the target locus, such as a local DNA structure, the presence of host protein binding sites, or DNA supercoiling. Dear students, it was all about today's lecture. Hope you have understood it well. See you with a new topic next time. Take care. Goodbye.